Here at learntobeatcancer.org, we are daily monitoring all of the latest insights, developments, research into all facets of cancer and reporting it out to you so that you don't have to do that hard work. Today we're going to take time to look at some really fascinating findings about how cancer cells can be better understood and potentially treated by looking into their epigenetic patterns. Epigenetic patterns have to do with how genes are switched on and off, rather than how genes are mutated. Researchers examined 24 different types of adult cancers and several pediatric cancers, and they were looking specifically at 720 genes that are controlled by these epigenetic switches. What they found was remarkable. Just by looking at the epigenetic patterns, they could separate tumors into distinct groups that predicted how patients would fare. For 10 of the adult cancers, including liver, lung, and brain cancers, these patterns were particularly meaningful. They could predict the patient's survival and disease progression. Even more interesting, when they mapped all these patterns together, tumor clusters not just by cancer type, but also by their location in the body. One of the most promising discovery was about potential new drug targets. Two specific groups of protein emerged as primarily and particularly important. And these help to control how DNA is packaged and how they activated the genes. These could be valuable targets for future cancer treatments. Research also revealed something surprising about the pediatric cases. While children's cancers typically had fewer genetic mutations than adult cancers, they showed similar epigenetic patterns to certain adult cancers, suggesting shared biological mechanisms that could be targeted for treatment. The key takeaway is this: when cancers looked similar genetically, their epigenetic differences can be what makes the significant difference. This opens up new possibilities for cancer treatment, particularly in developing drugs that target these epigenetic regulators rather than just targeting genetic mutations. While the study had some limitations, particularly in patient diversity, it represents a significant step forward in understanding how cancers differ at the epigenetic level and how we might better treat these in the future.